So, sorry, once again, let me try and go back a bit. Our lecture today is going to be on the function, the component and function of the adaptive immune system. So, the last time, the first lecture, we were able to look at some key features or characteristics of adaptive immune system, and that was when we look at adaptive immune system as being very specific. It is highly specific. It is going to target the specific antigen. For example, if it is parasite, if it is parasitic infection, adaptive immune system will make sure that the response is going to be meant specifically for parasitic clearance. And apart from being specific, the second one is the ability to retain some of these cells as part of the immunological memory. So memory cells, memory retention is key in adaptive immune response or immune system. So then the third one, which we are emphasized on, is versatility. Versatility. Okay. Microorganisms will put up different kind of antigen, and adaptive immune system has the ability to take care of diverse infection or infectious agents. Then the fourth one is self-tolerance, ability to be able to differentiate between self and non-self. If any immune system or immune response is not able to recognize or differentiate between cells that are made from the self and those ones that are foreign, that means it is going to treat every cell that comes as either part of self or as part of a non-self. If it is part of self, that means there will be tolerance. The immune system is going to tolerate the antigen to be in form of to be part of the system. So that means there won't be immunological response. That means it's not going to treat it like an antigen. But once the cell is able to treat it as cell, so it's going to recognize it as an antigen. And don't forget that I mentioned that one of the characteristics of regarding a substance as an antigen is that it must trigger an immune response. It must trigger an immune response, and it must be foreign. It must be recognized as foreign. Then the fifth one is the regulatory properties of adaptive immune system. So after everything, that is, after some memory cells must have been retained, after the intruders must have been taken care of, maybe he has recruited different cells to be able to fight against that kind of infection. So the adaptive immune system must have the ability to bring back the cell into normalcy, and that is part of the regulatory mechanism as being part of as being intrinsic in adaptive immune system. And the last one is that it has the ability to targeted destruction. It has the ability to target some pathogens. Okay, so whatever is going to be a kind of response, that means the response must be directly or must be directed to the certain pathogen. So, and that's what we learned in the first part of our discussion on adaptive um, immune system. Now today we are going to be concluding parts of this, and that is we want to look at the components. What are the components of adaptive immune system? What are the basic components of adaptive immune system? And last time in the earlier video, I was able to define them into two major components. The first one is called cell mediated, or what we call cellular immune response, cell mediated, and the second part is called the humoral immunity. So that is targeted at cell mediated can also be further divided into T lymphocytes, that is T cells, as well as this one has to do with humoral, that is antibody response. So this one concerns T cells. Okay, so and majorly when we look at, I told you that what produces the immune system arises from the hematopoietic cells or pluripotent cells of the hematopoietic cell, that is that will come from the blood stem cells, also regarded as the blood stem cells. And from that blood stem cells, this is further divided into myeloid stem cell. So under that myeloid, the first part is the red blood cell. The first part of myeloid cell stem cell is red blood cell, then followed by platelets, then followed by the third part, which is called the myeloblasts, So it is this myeloblasts that will further be differentiated into granulocytes. It is further divided or differentiated into what is known as granulocytes. So, and granulocytes are of three. These granulocytes, they are of these three different parts. So the first one is eosinophils, just like what we see in the innate immune system. Basophils. Each of these will take care of different types of antigen depending on the invading microorganisms. So don't forget that I said this is the blood stem cells. That is where the blood stem is being produced, and it is produced in the bone marrow. And the certain location of the bone marrow where it originates from is a hematopoietic cell. So this can be divided into two. I said myeloid stem cells, which comprises of a red blood cell, the platelets, and the myeloblasts. So myeloblasts will further be differentiated. This is going to be differentiated into granulocytes, and granulocytes consist EMB. That is, E is standing for eosinophils, N is standing for neutrophils, whereas B is standing for basophils. So out of these three, the neutrophils is the most abundant. It is the most abundant, and it is very, very mobile. Very, very mobile. In fact, it will be the first to get to the site of any infection. So the immune system always have, because it is most abundant, it is found almost everywhere in this blood cell. So it is most abundant, it is highly mobile, and it has a chemotatic potential. It has, that is, it can serve as an attractant, because it will always attract enemy to itself, because it is most abundant. It's just like when there is a infection by maybe armed robbers, and the numbers of security that are recruited to deal with just 10 armed robbers, they are like 1,000. You will see that once that happens, that means it is going to overwhelm the antigen. So neutrophils is the first to be recruited to get to the site of any infection. So because it is most abundant, it is highly mobile, and it has a chemotatic potential or chemotatic properties, because it will serve as an attractant. It attracts microorganism or antigen to itself. So it has that attractant, and it has the ability to swallow. It has the ability to swallow and or presents antigen, or present antigen to T-mediated cells. So we are going to see this. So the second arm is where you have the lymphoid cells. Is the second arm, the lymphoid stem cells. So the lymphoid stem cells will be divided into three as well. We have B lymphocytes, that is number one. B lymphocytes, that is where antibody is being produced. Then number two is the T lymphocytes. So then don't forget that we have cytotoxic cell. We have cytotoxic CTL. This is cytotoxic lymphocytes. Then we also have T helper cells. All these fall under T lymphocyte. Then the third is the natural killer cells. Okay, so that is when you now look at it properly. Now, if you look at it very, very critically, you will know that apart from the red blood cell and platelet, all this that is from here to this place, they are being regarded as white blood cells. So that is why if somebody asks you that you collect blood sample, that blood sample is made up of red blood cells, the platelets, and the white blood cell. So it is in that white blood, don't forget the function of white blood cell, that it is the part of cell that is going to protect, fight against invading organism. So, and in the ability to do that, That's why they are differentiated into different types of cells. So it is white blood cell that you found eosinophils, neutrophils. Some of them will be recruited as neutrophils, and some of them will be basophils, and some of them are going to be in form of B cells, T cells, and natural killer cell. And that's why that when they do what is called full blood count for you, so the number of each of these, the percentage of each of these will actually tell whether you are immunocompetent or you are not immunocompetent. For example, the neutrophil that is supposed to be the most abundant, if it is now found that it is another one that is more abundant than this one, that means once there is any infection or antigenic infection, that means the neutrophils will not be able to be recruited first to that site of infection. And don't forget that the ability, the immunocompetent ability of an host, or the immunocompetent ability of an host will depend on one, whether the type stroke the number that is being recruited first. So if, for example, if it's just like when you have 200 armed robbers that is invading a bank, and if the first set of people that will be recruited to that site of infection are security agents that do not have gun, do not have rifle. So even if, irrespective of the number, even if they don't have any harm, if they are not armed, that means that easily the armed robbers will overpower the security agents. Then the same thing with the immunological response. Whether somebody is immunologically competent will depend on the type of the cells that will be recruited and in their numbers, in their percentage. So you need to know that. But you should not forget that neutrophils is the most abundant, it is highly mobile, and it has chemotactic potential, that is ability to attract foreign antigen into them. It's just like I remember in those days that they normally tell you a story whereby the tortoise will always go and steal water. When they were digging the well, the tortoise was nowhere to be found. So it is just during the night when everybody must have slept, that is when tortoise will go and fetch water with all his children. At a point in time, they just discovered that somebody is already fetching their water. So they now decided to put a statue that will be sticky statue. So immediately that is going to be in form of human. So immediately the tortoise got there and he saw the standing statues. So what he did was, not knowing that it was already coated with sticky agents. So the first thing is he slapped the statue, the hand became gum, and like that until his whole body became so sticky. So the same thing is what neutrophil has, because they have chemotactic properties. So that means they have properties that can make antigen to get stick to them. So and once they have been able to get them stick, the body will be able to deal with it as anyhow. So that is that about the components. So now all these major components, we will now start picking them one after the other. We start picking them one after the other. Okay, don't forget that we are still talking about the adaptive immune system. So the first thing we are going to, the first cell that we are going to talk about, we will focus it on lymphocytes. Lymphocytes, okay? So lymphocytes originate from bone marrow. They have their origin in the bone marrow, don't forget, but they are divided into B cells as well as T cells. So they are divided into B cells, T cells, and Tregs. Okay, Tregs. So the second part of lymphocytes is what is called APC, antigen presenting cells. It's what is called APC. So the third component of lymphocyte is antibody. Bodies. So on what we call immunoglobulin. Okay, this is number one, which is mainly made up of B cells. Okay, and then followed by, then we have antibodies. There are 5. We have antigen. This is immunoglobulin. We have A, we have B, antigen B. We have IgM, we have IgD, and we have IgE. This basically divided into 5. So we are going to also devote some of these lectures to consider different types of immunoglobulin, and we will start considering picking them one after the other. We'll look at their components, we'll look at their functions, and we'll look at how they are able to do that and perform that. So this is number 1, this is number 2, number 3. Number 4 of lymphocytes is component is what is called the major histocompatibility complex, MHC, major histocompatibility cell. So these cells, there are 2 MHC, major histocompatibility complex. We have MHC1 and MHC2. MHC1, we always bind with CD4, and this one we always bind to CD8. So MHC1, MHC2. So MHC class 1 is found on the surface of CD4 cells, CD8 cells. MHC2 is found on the surface of CD8. So don't forget that whenever there is an infection, HIV infection, it is this CD4 that they will normally count. That's why they say if you want to diagnose if it is a viral infection, you have to go and look at the number of CD4 that the patient is having. So that is number 4. Number 5 under lymphocytes. Number 5 under lymphocytes is the cytokines. And what are cytokines? These are small molecules. They are small protein. I've defined as what cytokine are for you when we are considering the major component of the innate immune system. So cytokines, there are different types. We have TNF alpha, we have interferon, and we have interleukin. That is interleukin. We have interleukin 1, interleukin 2, interleukin 4, like that. So, but these are small proteins that are synthesized to deal with intracellular antigen, intracellular organism. So, don't forget that, especially they deal with intracellular. Cytokine are specialists for intracellular infection. That is, if the antigen is going to target the internal cells, internal part of cell. Then number two, they are also recruited to, in case there is their this is when you have viral infections, because most of the, you see, viral infection, don't forget the virus are intracellular. They are intracellular, and that's why cytokines are specialists in dealing with them. You see, I told you that adaptive immune system, they have they are very versatile. Their action is always specific. They have different they have different cells that will go and take care of different infection. So whenever the antigen or the infection is coming from viruses, or if it is a cancerous cell, cytokines are better. They are specialists in dealing with that. So major histocompatibility complex, you see, so the type of infection or infection that will now tell the types of cells that will be more recruited from the bone marrow. Because of what essence do you have? Because the cell, therefore, if immune response, they always work with prior experience. That's why they are delayed. They are delayed because they have a lot of, depending on the situation. So if it is what is going to involve antibodies, okay, the aspects that will produce more of antibodies will be produced. So it could be A, it could be antibody antigen B, it could be immunoglobulin B, it could be M, it could be, depending. So they are specialists. Adaptive immune response are specialists. They are specialists. Then this last one component of the adaptive immune system, the last one is the sorry, the sixth one, not the last one. We also have it up to 7. So the sixth one is the complement system. One thing you have to know at this point is that you will see that there are some of the components of adaptive immune system that you also found in the innate immune system. That's why they are network. We call them, they function in form of network. They are interwoven. So there are some cytokine is also part of innate immune system. Complement is also part of innate immune system, and more of these we have a kind of interconnectivity whereby they will interrelate or interact with each other. So just like when you are considering different types of security agencies in Nigeria, there are places whereby the military will invite the police. There are times that they work in hand with other security agencies like EFCC, Air Force, and so on like that. So depending on the situation. So, and the most effective and the most part of the adaptive immune system is memory cells. They have some cells that what they do most is they do what? They remember the insults from the pathogen and they store them in case there is reinfection. So all these are part of what we should know that and at every point in time, at every point in time of the adaptive immune response, memory cells, all of them, irrespective of the type of cell that is being recruited, all of them must have certain cells. In fact, the most abundant part of it is used as a memory cells. So if at the end of the day, if the recruits, if they recruit, okay, if from the bone marrow they now produce 90%. So it could be only 20% will be used in producing all this. The rest 70% will be used as memory cells. So that in case there is a reinfection, and that is why in vaccine production, any product of vaccine that cannot trigger the adaptive immune response, that means it is not going to be a good vaccine, except that vaccine is actually made for children, and that means it must trigger the innate components of the um, immune system. So, but if it is a vaccine that is made for adults, so that means it must, because what do we have as a vaccine? Vaccine is just a product of infection. The product of infection that we prepare system, that will prepare the immune system to be ready in case there is a what, and that's why they call it. That's the basic principle of what is called vaccination. That's what is called vaccination. Now, on the surface of, I told you that on the surface of CD8 cell, CD8 is regarded as cytotoxic cell, whereas CD4 is what? Helper. It's just helping. So we are going to see more of this by the time we actually go on. Don't forget that today, again, we have been able to look at the components of the adaptive system as they are produced from the bone marrow, and that is from that bone marrow, from the bone marrow, only produced from bone marrow. Other components, cellular components, some of them are also produced from the bone marrow. But the one that will be responsible for protecting of that will be immunological cells will emanate from the myeloid, which is from blood stem cells. So from that blood stem cells, there were two, which could be myeloid stem cells. It is from that myeloid stem cell we have the red blood cells, we have the platelets, and the third part, which is a myeloblast. That one will differentiate into granulocytes, and the granulocytes will produce three types of cells. We have eosinophils, we have neutrophils, and we have basophils. I've told you that out of these three, the neutrophils are the most abundant. They are the first to go and to encounter and challenge antigen, because they have attractant properties. They are chemotactic in nature. So then the other arm of the blood stem cell is lymphoid stem cells, which will produce lymphoblasts. These lymphoblasts could be of B cells, it could be of T lymphocytes, as well as natural killer cells. But you should know that the myeloblast as well as lymphoid stem cell, or what we call lymphoblasts, they are the ones that are called the white blood cells. They are called the white blood cells. So that's why if somebody is asking you that inside the blood what do we have? There are three major components of the blood. The first one is red blood cell, the second one is platelet, and the third component is white blood cell. And these white blood cells, they are of different they have different types. They have different types. So, and the other arm, which is lymphocytes, which is lymphocytes, they are also divided into components, different components of lymphocytes. There are some that we further differentiate into B cells. There are some that will differentiate into T lymphocyte cells. These T lymphocyte cells, that's where you have T cell. That's where you have CD4 and CD8, cytotoxic or T helper cells. It's a type of T cells. Then apart from that, we have you see the difference between helper cells, the difference between T helper cells and cytotoxic is that the T helper, that's from the name helper, they are the one that will just help to coordinate. They will only coordinate for another. It's just like if somebody cannot arrest the criminal and the person just gets immobilized. Yes, that's the right word. We now immobilize. The T helper cell will immobilize an antigen. They will not allow them to go. They will arrest them pending the time that the very competent or more specific cell is being recruited, and that's why it is on the surface of the CD4 or CD8. So that one will just go and present the antigen. We present them as antigen. Okay. So, but the cytotoxic cell, they have the ability to kill outrightly. They have the ability to kill. You query them. So that's the difference between cytotoxic cell and the T lymphocyte. Because once they recognize that this is an antigen, they will deal with them directly. To directly deal with them. But what they do is to just immobilize them. Pending the time that some other one will be. So then the third aspect, which is called the T-regs. So you see, if there is nothing to regulate the ability or the function of either cytotoxic or T helper cell, that means there's going to be problem at the end of the day, because more energy will be wasted, and and before you know it, the cell will begin to fight against itself. It will begin to fight against it, because it's just like when you have invited soldiers to come and help you to deal with your with criminals. At any point in time, by the time you start, the soldiers deal with your criminal. That means you are the one that will start supplying them with food. So if you are not so careful at any point in time, you yourself, you are not safe again. So that is the function of the T-reg. So it is the one that will bring back, will maintain, will stabilize, will equilibrate the cytotoxic as well as the T. Okay, then this second aspect, which is antigen presenting cell. This one, they don't have any other thing. They don't have any other function. Rather to present on the surface, on their surface, they will go and present the signal of the enemy. They will go and present the signal of the enemy, that this is the signal. It's just like somebody that they don't have any weapon. ABC, they don't have any weapon. The only thing they have is that once they have once they see any antigen, their own is just to take the photograph. Just take the photograph of the enemy, and they go and paste that photograph on their own surface. so that everybody will know that these are the invading or antigen that have invaded the system. So you will see that all of these, including those ones that arrest, including those ones that immobilize, including those ones that deal with them directly, including those ones that will just take photograph, they are called the antigen presenting cell. These ones are photographers of the immune system, because whatever might be, so they will just go and display it on their own surface. They are antigen presenting cells. Okay, and example of antigen presenting cells, they are there are different types. Dendritic cells also perform the role of antigen presenting. Macrophages, they also perform the role of antigen presenting, as well as B cells, as well as B cells. So don't forget that the dendritic cells are very, very, very important. The dendritic cells, they are very, very important. The dendritic cells are very important. One of the reasons why they are very important is that they are the one that will go and stimulate. They stimulate naive cells. They are the one that will stimulate the naive cells. Don't forget that I told you that there are some cells, they do not have any prior experience. They are just like virgin. They are just like virgin that do not know anything at all. They don't have any bad experience. They don't have anybody that has ever broken their heart. So, but the dendritic cells, they are the ones that will go and prepare all the naive cells. They will go and prepare all the virgins that have no prior experience. That will tell them that, see, there are going to be problems. And anytime you have that problem, this is what you should do. So the dendritic cells is part of antigen presenting cells, and that actually stimulates the naive cells. Okay, and the third part, which is antibody, or what you call immunoglobulin. The first one, which is most common in the blood, and it is found in other, not only in the blood, it is also found in the other fluid of the body, is the antigen G. Antigen G. Sorry, this is not bio. G. Antigen G. I'm sorry, immunoglobulin G, that is IgG, which is the most abundant, because this one will provide a long term. We are going to see, we are going to dedicate for this, a video will be dedicated for immunoglobulin. So at that point, we are going to see them more. So the major histocompatibility complex is parts. These are a very important part of the very important part of the antigen presenting cell, that is the adaptive immune system. And because what they do is that they present um, all nucleated cells, MHC class 1 present all, they are present, sorry, they are present on all, they are found in all nucleated cells. They are found in all nucleated cells. They are found in all nucleated cells. And all nucleated cells, as well as cytotoxic cells. All nucleated cells, as well as also found in the cytotoxic cells. Okay. So I said the major histocompatibility complex, they are of two types. MHC, they are of two types. Class 1 and class 2. These class 1 are present They are present in all nucleated cells of the body. That is, all cells that contain nucleus in the body, and those one that perform the role of cytotoxic cells, they are found in them. Okay, they are present in all them. Then that makes them to be internal. That make their function to be what? Internal. But the class two, which are said to be external, they are present on all T helper cells, okay, and they are found in all cells that are external. So what normally happen is that there is something we call TCR. TCR is T cell receptor. All these one are very very important in the sign of effective vaccine for any ailment and by the time we start considering bioinformatics vaccine design, we are going to see more of this T cell receptor. So there must be interaction between MHC, either class one or class two. There must be a kind of interaction between MHC1 or MHC2 and what is called the T cell receptor. It is like they are. It is just like cap that is fitted. So it is a cap that must be fitted into MHC1. So there must be a regular contact or there must be a contact before that. So then we also mentioned something about cytokines. And I told you that cytokines are just small proteins that are being recruited for to deal with intracellular organisms, especially viruses, as well as when we have infection that will lead to cancer. So cytokines are better recruited in that occasion. So, and I gave you different types of cytokines. I mentioned TNF alpha or TNF gamma, as well as interferon. Some of them are grouped according to their interferon. And we have those that are interleukin 1, interleukin 2, interleukin 4, interleukin 10, and like that. So all these are very, very important in the design of a vaccine. We are going to see more of this and how they are used in the design of a vaccine by the time we go. So then after that, we now talk about complement system. Don't forget that particularly, I told you that complements are made up of different proteins that complement the work of other members of the immune system, not only in adaptive system, also in innate immune system. Just like use. So they are just set of complement system are just like 20, I think they are around 27. So once one is activated, one is just activated, this other one will send signal. It's just like when you line up empty plastic, when you line them up. So when you kick one, that one will now send signal to others, and you will see that others too will also just scatter at the end of the day. So that is complement system. Complement system are made up of more than 27 proteins that once one is activated, is going to cascade, is going to activate the other part. And I told you the last part of the adaptive immune system is memory cell. So all this are long-lived. Memory cells are long-lived. Even when the infection, if when the infection has been cleared and the body has actually been brought to normal, memory cells will retain, they will retain, they will retain the memory of the pathogen for a long time, for a long time. In fact, sometimes for a lifetime. So that is why you have some you have some immunization or vaccine that will last only for 2 years. Some will be for 10 years. Some will be for life. So depending on the one that is being used. So conclusively, you will know that adaptation is the most efficient, is the most effective, it is the most organized part of the immune system. In fact, if what we have is only innate immune system, our body or the body of an organism will have been overwhelmed, because if nothing, if the innate immune system does not have anything like memory, we don't have any memory. Once they have cleared the infection, they do not have anyone that is going to. And you can see, just like I told you can see how all these cells, how they work intricately, how they work hand in hand in form of a network. And it is because of this that the body of the host is able to protect the host from or fight against different types of infection. Just like I told you, adaptive immune system is highly specialized. It's specific, not only specific, it is highly specialized, it is highly specific, it is able to differentiate between self and non-self. It has the ability to deal with different types of pathogen at different types of cells. You can see innate immune system cannot actually deal with cancer. Most of them cannot deal with they cannot deal with viruses, and that's why you have more in children. The small children is more is fascination than just the one that is natural from adaptive immune system. So this one is able, it has